Okay, welcome to chapter 5.3. We're going to be talking more about bacterial cell walls and their outer layers in this lecture. So we're going to go over gram positives and gram negatives again, and we're going to learn about one that we haven't really talked about up to this point. Now, before we start into the section for 5.3, let's do a review question. So, which of the following is true of passive diffusion? It occurs with the concentration gradient. It never involves a protein transporter. C, it occurs against the concentration gradient. Or D, it always involves a protein transporter. Okay, so pause the video and find the answer to this question. Okay, so the answer to this question is A, it occurs with the concentration gradient. Remember, passive transport or passive diffusion, passive transport does not use energy, so it uses a concentration gradient. So concentration gradients like to flow naturally from a high concentration to a low concentration. Okay, so which is what A is saying. Now we can immediately eliminate two answers from this question. Okay, so we know that transporters are involved, okay, but B and D make some absolute statements. It's either they're never used or they're always used. So no, we know in the case of diffusion that it can go with or without the transporter. Okay, so that automatically eliminates B and D because if we can go directly through the membrane, it's not always going to use one. And if we use a transporter, it's never going to fall. It's not going to fall underneath B because it's using a transporter, so it's not going to be never. Okay, so that leaves A and C. So it's either it occurs with or occurs against. So no passive transport or passive diffusion is going to be A. Now this question could be rewritten to which of the following is true of active transport. And again, B and D aren't going to come into play, okay, because we have a lot of active transport that does use transporters, but we have bulk transport like endocytosis, so phagocytosis, that isn't going to use a transporter. It's going to use the actual membrane to make a vesicle when it brings something across. Okay, so it's either going to be between A and C, but with active, it's going to go against the concentration because we're going to use energy to go from low to high. So this is a good question to know because it's one that could show up on the next exam. Okay, section 5.3, we're going to talk all about bacterial cell walls some more. Okay, and we're going to talk about those outer layers that are by the cell wall. Okay, so objectives for this section is you'll be able to describe the cell wall structure and explain how it protects bacteria from osmotic shock. You'll be able to explain the function of gram-positive cell walls and teichoic acids, and you'll be able to explain the function of gram-negatives and gram-negative outer membrane, the LPS, and something called the periplasm. Okay, so we've talked quite a lot about cell walls. We talked about the fact that it's made of these glycan chains with these peptide cross-linkages, okay? And we talked about how it's a cage for bacterial cells that are going to help it survive its environment. Okay, so it's either going to make it so that if it's in a hypertonic condition, when all the water rushes out, it's going to make it so that the cell doesn't shrink and implode upon itself. And if it's in a hypotonic environment, it's going to absorb water, but it's not the cell wall is going to act like it's a cage that's not going to allow it to expand too much until it bursts. Okay, what we really haven't talked about is the fact that the cell wall is one single molecule. It's all connected. Okay, so in order to make this cage, all these glycan chains, all these peptide cross-bridges are connected to each other, so it makes it one molecule. Okay, so almost all bacteria have cell walls. About 90% of all bacteria have cell walls. So we've talked about that 10%, like the mycoplasms and the ureaplasms. One I always forget are the chlamydias. These are all cells that have lost their cell walls. Most of the time, mycoplasms and chlamydias have lost their cell walls because they don't need them. Okay, they're going to be what are called intracellular pathogens, meaning they have to have a host cell. They have to live inside other cells to survive, which is one of the reasons why they don't need their cell walls, because inside the cell is pretty constant, right? One of the properties of life that we learn in biology is homeostasis, so a constant internal condition. So both mycoplasms and chlamydias don't really have to worry about these osmotic pressures, so hypertonic, hypotonic conditions, because everything inside the cell is isotonic. It doesn't need that protection of the cage that is the cell wall. Okay, so we're going to talk a little bit about this peptidoglycan in a little bit more detail. We know we have these glycan chains. These glycan chains are actually made of disaccharides, okay, that are going to be connected in a polymer. Okay, and then, of course, the peptide, these amino acid sequences, are going to connect each of these glycan chains. Okay, so don't freak out on me. Okay, there's a lot of detail on this page. You don't need to memorize this. You don't need to know. But it's good to understand that these cell walls, as simple as we talk about them, they are a little bit more complex. So let's talk about these glycan chains. These glycan chains are a disaccharide. In fact, they're made of NAG and NAM. NAG stands for N-acetylglucosamine, while NAM stands for N-acetylmuramic acid. Don't need to memorize these. So these are two saccharides that are joined together by a glycosidic bond. Okay, so they're always in this set of two. Now, the cross linkage, the peptide cross bridge, is always going to be attached to NAM in this place. Okay, so here M is NAM, G is NAG, so you can see them alternating in this polymer. Okay, and each time we have these cross bridges, we have the same amino acids. We have D-alanine, we have glutamic acid, L-alanine. Okay, and then we have this one that is not quite an amino acid anymore because it has made this bridge. So we have N-diaminopylic acid. Okay, so this cross bridge is going to be between this diaminopylic acid and the D-alanine and a connecting chain. Okay, the difference between these two, they're just going in opposite directions. Okay, so this is going to be where they're linked. This is what's going to make it a single molecule. Okay, now we have a set of antibiotics that are going to target different parts of the cell wall. Okay, so this little guy right here, let me turn my thing on. Okay, so we have our diaminopylic acid, we have our D-alanine, we have our cross bridge right here. Okay, so this bond right here, okay, is an important drug target. In fact, penicillin is part of a drug class of antibiotics called the beta-lactams. Okay, beta-lactams are going to target the cell wall. Okay, so penicillin itself is going to disrupt this bond right here. We'll talk more about that in a little bit more detail on the next slide. Okay, we have other beta-lactams like vancomycin that is going to affect this D-alanine right here, and it's going to make it so that this comes off and the cross bridge has nowhere to bind to connect them. Okay, anytime you don't have this cross bridge linkage, we have a hole. We have an unsteady part of the cell wall. Okay, remember antibiotics, they punch holes in the membrane and in the cell wall. Because that we can make these holes here or here, this is going to affect the protective properties of the cell wall, because now we have a weakness, we have a weak link, like literally we have a weak link. Okay, so when the cell is in these hypertonic and hypotonic environments, this is not going to keep it steady. Okay, we're going to have this hole, and it's going to cause things to leak out, and the cell's going to go into osmotic shock. Okay, so that's one way that we can poke holes in the cell membrane. Okay, as I said, penicillin is part of a drug class called the beta-lactams. Okay, and all these beta-lactams are going to target other really important pieces of the cell wall. Now, let's talk about penicillin in general. Okay, so anytime we have a disruption in the cell wall, it's going to make the bacterial cell more susceptible to osmotic pressure and changes in it. So changes in hypertonic versus hypotonic conditions. Okay, and it's not going to be there to prevent the cell from bursting, especially in a hypotonic condition. Okay, so because the cell walls are made up of this peptidoglycan, they make a really great antibiotic target because we don't have that. Okay, so penicillin is going, and other beta lactams are going to disrupt the synthesis of peptidoglycan. Okay, so penicillin itself is going to disrupt this cross-link. Okay, so there is an enzyme called transpeptidase, and this is going to be the protein that goes in and connects that cross-link to that D-alanine and makes it one continuous, not specific, continuous molecule. So what penicillin does is it comes and it binds to the place on the enzyme where it normally binds that D-alanine. Okay, so it's going to bind here, and when it binds to the transpeptidase, this enzyme, it's effectively going to turn the enzyme off. Okay, and then that linkage can't be made, which makes the cell wall weaker, makes it the cell more susceptible to osmotic shock. Okay, so different beta lactams are going to have different mechanisms of action, so they're going to affect different things. Okay, but penicillin itself is going to bind to this transpeptidase. Okay, and then that link is no longer going to be made. Okay, so as I said, penicillin, it is used still to treat things like pneumonia, strep throat, syphilis, and gonorrhea. So vancomycin, which is another type of beta-lactam, so it just targets a different part of the cell wall. Okay, it's going to be, hopefully I can say this today, is used to target the Clostridiales. You can read it on the slide. That species of bacteria, the C. diff, Clostridium difficile, and different staphylococci species. Okay, so different antibiotics are going to be useful on specific species. Okay, but they all disrupt the cell wall in some sort of way if they're a beta lactam. Okay, unfortunately for us, some bacterial cells have evolved to be resistant to beta lactams. Okay, so these beta lactams are going to disrupt the cell wall. So some of these bacterial cells have evolved to be resistant to these beta lactams. In fact, penicillin, a lot of bacterial species are now resistant to it. We don't use it a lot in treatment anymore because of it. Okay, because penicillin is a beta lactam, all beta lactams have a very similar structure. Okay, so they all have this ring structure that is part of the antibiotic. So bacteria have evolved and now have an enzyme called bacterial beta lactamase. So what beta lactamase does, its whole job is to bind beta lactams like penicillin and break them. Okay, so it's going to break them, and then penicillin is no longer effective and can no longer bind to the enzyme, which is going to then prevent that cross-linkage from forming. Okay, so bacterial beta lactamase is going to stop this process here, and it's going to allow for cell wall synthesis. Okay, it's going to allow for this peptide cross-link to actually happen. Okay, so we're starting to see a whole lot of bacterial cells become more and more resistant to the beta lactams. This is another reason why that we usually give you an antibiotic cocktail, so 2 or 3 different antibiotics at once, because we don't know what species of these pathogens you have, and if they are going to be resistant to our beta lactams like vancomycin and penicillin. Okay, so let's talk about the outer layers. So we talked about 2 cell wall types. Okay, remember 90% of all bacteria have cell walls, but we've specifically talked about 2 types of bacteria that we classify based on their peptidoglycan layers. we have gram-positives and gram-negatives. So we haven't really talked about phyla. These are different categories of bacteria. So gram-positives are going to be a part of the phylum Firmicutes. Okay, so this is going to include species like Bacillus anthracis and Streptococcus pyogenes. Okay, gram-positives all have some very consistent characteristics that happens in all gram-positive cells. Okay, they're going to have a thick cell wall, which means they're going to have multiple layers of peptidoglycan. And we're going to talk about some of the other the other characteristics they have, and we're going to look at how we can start to identify them versus gram-negatives without a gram stain. So this is actually going to be a big part of your next exam. So this is a good part to pay attention to. Okay, gram-negatives, on the other hand, are part of the proteobacteria phyla, and they are the opposite of gram-positives. They have thin cell walls, they have a single layer of peptidoglycan, and one of the other differences is gram-positives only have one cell membrane. Gram-negatives are going to have two, so they're going to have an inner membrane and an outer membrane. Okay, so different gram-negative species that we are worried about are Pseudomonas aeruginosa and a good old E. coli, so Escherichia coli. Okay, so let's talk about these gram-positives and gram-negatives in more detail, and let's learn how to tell them apart. Okay, so the easiest way, of course, to tell a gram-positive and a gram-negative apart is just to use microscopy, right? Use the gram stain. Okay, gram-positives are going to be purple because of the crystal violet that's trapped in their peptidoglycan layers. Okay, gram-negatives are going to be pink because they doesn't trap the crystal violet. Okay, so gram-positive cells. So this micrograph here and this schematic are really important to know for the next exam. Okay, I would be able to look at this schematic and say, ooh, this is a gram-positive. Okay, so let's learn about what makes it a gram-positive. So let's start with the micrograph. The micrograph is pretty easy. So in a micrograph, we're only going to have one dark line. Okay, this is the cell membrane. Now, if you look here, we can see this dark fuzzy area. This is actually the cell wall of the gram-positive, so it's going to be that peptidoglycan layer. And we can tell that it's kind of a thick guy. Like it takes up more space than the actual cell wall. Okay, so this single membrane in a micrograph indicates that it's a gram-positive. You need to know this. Be able to identify this on a test. Okay, so what does that look like? Okay, so we're going to have our cell membrane, like in our membrane proteins. We're going to have our cell wall. Okay, we're going to have our multiple layers of peptidoglycan. Now, what we haven't really talked about is that there's another component of the peptidoglycan layer, and it's called teichoic acid. Okay, the teichoic acid is going to react with that crystal violet, and it's going to help it stick in this peptidoglycan layer, like the mordant iodine is going to let it do once it creates that complex. Okay, this teichoic acid isn't going to let that crystal violet out. Okay, the thing is with teichoic acids in gram-positives is teichoic acid is something that our immune system recognizes. Okay, so it's really easy to identify gram-positives this way. Okay, and on the outside of our peptidoglycan layer, we're going to have these glycosyl chains. So these are going to be polysaccharide chains. Okay, and just a reminder, because the crystal violet is going to stick in the peptidoglycan layers, it's going to be purple in the gram stain. Okay, so what we're looking for when we look at these schematics, we're looking for multiple peptidoglycan layers, multiple NAGs and NAMs, and we're going to only look for one membrane. Okay, and for teichoic acid if it's labeled. Okay, so our gram-negative cell wall is much thinner. Like we're going to have a single layer or two layers, going to be very thin. Okay, so that's the main defining characteristic. But one of the biggest differences between gram-positives and gram-negatives is the fact that gram-negatives have two membranes. So they're going to have an inner membrane. In fact, this inner membrane is going to be the cell membrane or the plasma membrane. Okay, this is the one that we talk about when we're talking about transport. Okay, and then we're going to have this outer membrane. Okay, the peptidoglycan layer, the cell wall is going to be in between the two membranes. In fact, the peptidoglycan is found in what we call the periplasm, or the periplasmic space. I would know periplasm for the next exam. Okay, we haven't really talked about what we call this space in between membranes, but in gram negatives it's called the periplasm. So in the outer membrane, we have what we call a lipopolysaccharide embedded in it. And we're going to talk more about the LPS layer here on the next slide. Okay, but if we're looking at this schematic, we're going to look for the two membranes, and we're going to look for this peptidoglycan layer, and we're going to look for the LPS. Okay, and if we're looking at the micrograph, we're going to look for two membranes here. Okay, because you can kind of see between the two membranes the peptidoglycan layer, but it's so thin it's not as easy to see as on the previous gram-positive slide. Okay, this LPS layer is really important because this is what makes bacteria like Salmonella species and E. coli make us sick. In fact, this outer membrane is part of the cellular envelope. This outer membrane is their defense structure. This is their first line of defense, and it can be toxigenic, meaning it can make something toxic. Okay, so this LPS layer is what's going to make us sick, and we're going to talk about what specifically in the LPS layer makes us sick. Okay, and just a reminder, because this has the thin peptidoglycan layer, gram-negative cells are going to stain pink and not purple. Okay, so let's talk about these outer layers, the outer part of this gram-negative envelope. Okay, one of the most important parts of this is the LPS layer, that lipopolysaccharide layer. Okay, so it's made up of different parts. Okay, so we have our LPS chains, but these are attached to what is called lipid A. So lipid A is going to be embedded in that outer membrane. Okay, so when gram-negative cells are intact, meaning that we haven't broken them in the process of treating them, these cells aren't toxic because lipid A is found in the membrane. Okay, so sometimes when you have a infection that is caused by a gram-negative, treating it with antibiotics can actually make you sick. And that's because our antibiotics are going to punch holes in this membrane layer. And what's going to happen is it's going to break it open, and it's going to start to release this lipid A from this outer membrane. So when we talk about LPS layer, the part that's toxic, so the part that acts as an endotoxin is what we call it, is this part that's embedded in this outer membrane, this lipid A. This is what makes you sick. Okay, so sometimes treating the bacterial infection can actually make us sicker, and that's because when we break this bacterial cell, it's going to release that lipid A, and that's going to be what makes us sick. Okay, so when the antibiotic punches a hole in the membrane, okay, this bacterial cell is going to kind of send like an SOS, and that signal is going to cause lipid A to be released from this outer membrane. So lipid A is going to interact with your immune system cells, and it's going to ramp it up. It's going to overstimulate the host response. Okay, and when this happens, it's going to cause you to go into toxic shock. Okay, but the thing is, this LPS layer is also very slick. Okay, it kind of has this mucus slick feel to it. Okay, this LPS layer is also going to help gram-negatives evade your immune system, because this LPS layer, because it's slick and mucousy, is going to give it so our neutrophils, our white blood cells, can't get a good hold onto those gram-negatives. They're going to slip and they're not going to be able to phagocytose them, and the bacterial cell is going to be able to get away and to keep causing an infection. Okay, so the LPS layer is very, very important. Okay, so we talked about the periplasm. So if we look here, we have our inner membrane or cell membrane. We have our thin layer of peptidoglycan. We actually connect both outer layers. They're not just kind of free floating. They're connected to each other. So this peptidoglycan layer is going to have a polypeptide. So we have our alanine, our diamino, the acid, I cannot remember it without seeing it, our glucanic acid and the other alanine. Okay, so these are going to be attached to what is called a murine lipoprotein, and this is going to keep the outer membrane connected to the inner membrane. Okay, and this space between the two is called the periplasm. You need to know that. In fact, you're going to have to be able to label one of these schematics in an exam. Okay, so remember, when we're looking for gram negatives, we're looking for the two membranes, the thin layer of peptidoglycan, and the LPS when we're trying to identify these from a picture or a micrograph. Okay, these are the two things side by side. Okay, you need to be able to tell the difference between these two. This is a large part of your next exam. I think about 10 to 15 points. So 15% of your next exam. That is quite the amount. Okay, so that's gram positives and gram negatives. So one of the things that we're going to talk about last in this section is mycobacteria. Okay, so mycobacteria have cell walls. They have peptidoglycan, but they have a different outer layer than both gram positives and gram negatives. In fact, mycobacteria can't be gram stained. And the reason they can't be gram stained is that they have this waxy coating. Okay, so they have this layer of lipids and mycolic acids on the outside of their cell wall. Okay, these mycolic acids and peptidoglycan are going to make it so that it has this waxy, rigid, hard outer layer. Okay, this is going to be beneficial for the bacteria, and also it's going to cause some interesting things to happen with bacteria. But this mycolic acid layer, this waxy layer, is what makes it so it's not able to gram stain. In fact, if we want to stain mycobacteria, we're actually going to use a special stain called a acid-fast stain. So we're going to heat up the bacteria, soften that waxy outer coating, and apply malachite green and get it to go into that waxy layer in order to image and stain that cell. Okay, this waxy coating is also a little bit detrimental to the bacteria. It's going to cause the bacteria to grow very slowly, but just like we can't gram stain it, and it's really hard to stain these type of bacteria, it also helps them and protects them from the antibiotics. So antibiotics can't penetrate that thick waxy layer. Okay, so mycobacteria have a unique envelope compared to both gram positives and gram negatives, which both have a unique envelope, but they're similar enough that we can group them together. Okay, so mycobacteria are going to have a few human pathogens. Two of the ones that we talk about the most are both Mycobacterium tuberculosis. This is from our first case study, remember? The student at the high school who got tuberculosis, and Mycobacterium leprae. Now, if you don't know, Mycobacterium leprae is the bacteria that is going to cause leprosy. So the last thing we're going to talk about is a case history called A Rash Reveals an Ancient Disease. So we have Naranjan, and he lives in India, and specifically he lives in Bihar. Now, one day, Naranjan started to develop lesions on his face and lips. Okay, so these lesions are these little pockets of fluid right here. Okay, so these lesions are very indicative of leprosy. Okay, in fact, it's not the bacterial infection that's causing these lesions, it's actually your immune system's response to the leprosy that's going to cause these lesions. Okay, so back in the olden days, people with leprosy, lepers, were kind of thrust out of society. They weren't allowed to interact with people because it was commonly believed that if you touched somebody with leprosy, you would get leprosy yourself. Now, only about 5% of the people who are exposed to leprosy actually get the disease, and it takes a long time to develop it. And it's not just by touching, you have to actually touch their nasal secretions. So like if somebody with leprosy sneezes on you, you have a higher chance of getting leprosy than just touching their face or touching their lesions. Okay, because Mycobacterium leprae, so the bacterium that causes leprosy, is a mycobacterium. It has that waxy layer of mycolic acid and peptidoglycan on the outside. So it takes a long time for this bacteria to grow. In fact, a lot of people will be exposed to the disease, so exposed to the pathogen, but won't actually get the disease to a year or two later because it's so slow growing. In fact, it is so slow growing. For us to grow it in the lab in ideal conditions, it takes us up to 6 weeks to grow it. So this is not one of those tests, one of these diseases that is going to be proven with a with a stain, or sorry, not a stain, with a quick test of growing it on the petri dish and in samples. Okay. In fact, in order to detect this bacterium, what's going to happen is they're going to take some skin scrapings of the lesions, and then they're going to acid fast stain these. Okay, so they're going to heat it that's going to allow the stain to penetrate that waxy mycolic acid coating. Okay, so this is what it looks like underneath the microscope. Now, one of the things that is interesting about mycobacterium is they like to grow in cooler temperatures. Okay, so if you look here at this guy's face, there's some places of his face that don't have as many lesions as others. Okay, so if we look at the thermal image here, we can tell that his forehead right here, his T area is what it's called, his eyes, and around his mouth and his neck are more warm than, say, his nose, the top of his forehead, and his cheeks. Okay, so these areas of blue and green are going to be where the bacterium is going to want to grow. So if you look at his face, we see a lot more on his cheeks, his nose, his upper forehead, and his bottom of his chin and sides of his neck. And of course, the rest of his body, like your extremities, so like your arms and your legs, are going to be cooler than the trunk of your body, your torso. Okay, so he's going to see a lot more lesions on his arm and his legs for the same reason. Okay, so fortunately for us today, leprosy is very, very curable. It just takes a while. So back in the day, because people with leprosy were cast out of society, some places like Hawaii, the king of Hawaii in the 1800s, when they started to interact more with different traders from Europe and America, they started to bring diseases over that they hadn't seen in Hawaii, and one of them was leprosy. So to prevent the spread of leprosy, what the king or the governor of Hawaii did is he designated an island for these people with leprosy to go to, and he made a leper colony. In fact, this leper colony was fully functional for over 100 years. Okay, so it was into the 1950s when people were still being put onto this leper colony. Okay, so as I said, leprosy is curable now, but it takes about 2 years of pretty strong antibiotics to get rid of the bacteria. And part of that is because it's slow growing, it has that waxy coating that's going to protect it from those antibiotics. So it's a slow process, but we can cure it. Okay, that is 5.3. We talked about the cell wall, we talked about gram positives, talked about gram negatives, and we talked about mycobacteria. I would know the distinguishing features of all the cell envelopes, so cell wall, membrane, and any of the outer layers for these 3 types of bacteria. I would know that mycobacteria uses the acid fast stain to stain it. And I would know that the difference between gram positives and gram negatives is that mycolic acid peptidoglycan layer, that waxy outer coating. Okay, before we end this video, let's do some review questions. So review question number 2. You are investigating the cause of an outbreak of food poisoning. The culprit contains peptidoglycan and lipopolysaccharide. Which of the following organisms could be the cause? Okay, Staphylococcus aureus, a gram-positive bacterium. E. coli, a gram-negative bacterium. Aspergillus fumigatus, a fungi. Or D, any of the above organisms could be the cause. Okay, so take a second, pause the video, and see if you can't find the answer. Okay, the answer to this is B. Okay, so let's look at the information that the problem gives us. So peptidoglycan, this tells us it has a cell wall. Well, we can immediately eliminate C because funguses are eukaryotic. They don't have a cell wall. They don't have peptidoglycan. So C is automatically off the docket. Now, if C is off the docket, D can't be true either, because if it doesn't have peptidoglycan, it's not causing this disease. So C and D are out, which leaves us between A and B, our gram-positive and our gram-negative. Now, they both have peptidoglycan, so what's the defining feature here? And that's the lipopolysaccharide part of this question. Okay, so which one has lipopolysaccharide? The LPS layer, and that's the gram-negative, so the answer is B, E. coli. Okay, so review question number 3. Which of the following is a characteristic of a gram-positive cell wall? A, waxy due to the presence of mycolic acid. B, the outer membrane with lipopolysaccharide. C, thick with several layers of peptidoglycan. Or D, a periplasmic space between membrane and peptidoglycan. Okay, so pause the video and find the answer. Okay, this is actually a great question because it can be easily reworded to go after the other two types of bacteria we talked about in this lecture. So the answer for this one is C, thick with several layers of peptidoglycan. That's the defining feature of gram-positives. Now, one of the things that makes this really interesting is we can put in we could put in gram-negative or mycobacterium in for gram-positive, and this question would still work. Okay, so if this was for a gram-negative, we'd have B, outer membrane of lipopolysaccharide, and D, the periplasmic space between membrane and peptidoglycan. These would be correct if this was a gram-negative cell wall. Okay, if it was asking for a mycobacterium cell wall, it'd be A because of the mycolic acid. Okay, so this is a good question to know and all its iterations. Okay, so the last review question number 4, asks which compounds are found in the outer cell layers of Mycobacterium tuberculosis, but not in most other kinds of bacteria. Say A, teichoic acids, B, S-layer proteins, C, murine lipoprotein, or D, mycolic acids. So take a second, pause the video, and try to find the answer. Okay, the answer to this question is D, mycolic acid, right? Mycobacterium, mycolic acid, they're very similar to each other. So this is D. Mycobacterium are the only ones with these mycolic acid outer layers. Okay, so let's look at the other one, teichoic acid. This is in gram-positives. Okay, they're in that thick peptidoglycan layer. So S-layer proteins, we haven't really talked about them. They do show up in our images. They're going to be on the outside of that peptidoglycan layer or the outside of that outer membrane for gram-negatives. This is where that outer layer, that capsule is going to attach to. Urine lipoprotein, this is in gram-negatives. It's part of that periplasmic space. So it's going to connect the two membranes together. And obviously D is the answer. Okay, so that is all for 5.3. We'll see you for the video in 5.4.